this seminar on prostate cancer. We are very happy to have the opportunity to have two excellent speakers today. And I think that the topic of prostate cancer is very important because prostate cancer is one of the leading cause of male death in Western developed countries. The comprehension of the molecular mechanism of the onset and progression of prostate cancer may help to develop more appropriately diagnostic and more appropriately and maybe personalized clinical intervention. So today we have a special guest, Professor Vieira Ana Filipa, is right? She comes from the Istituto Politecnico do Porto, Portugal. She has won an Erasmus grant to experience a teaching mobility through visiting the Anatomia e Histologia Patologica of Trieste. She holds a PhD in molecular pathology and genetics, and she has been teacher of oncobiology, molecular biology, immunohistochemistry, cell culture proceedings, and histological techniques. It's right. Nowadays, she works as a researcher at the Cancer Epigenetic Group Research Center of Portuguese Oncology Institute at Porto. The Erasmus project is particularly important for us because many students of the biomedical laboratory technician course have the opportunity to visit for training different institutions like that of Portugal, from which Filipa Vieira comes. Professor Zanconati sustained 3 bilateral agreements with Istituto Politecnico do Porto, Portogallo, Universidad de León, Spain, Universitat de Medicinas y Farmacia Târgu Mureș, Romania. From 2013, Anatomia Histologia Patologica of Trieste has admitted 14 Erasmus students for training, 2 students in Erasmus studies from Romania and Spain, and 2 teachers from Istituto Politecnico do Porto. We hope that this opportunity will be extended over the time. Let me give you some information about the lectures of this afternoon. The first speaker will be our guest, Professor Filipa Vieira, with the lecture Histone Modification in Prostate Cancer. Then the second speaker will be Dr. Alessandra Bosutti, who will present a lecture on the eukaryotic elongation factor, EF1A1 and EF1A2, as new potential molecular targets and biomarkers in prostate cancer. Alessandra Bosutti is a biologist with a PhD in molecular medicine of the University of Trieste. She has moved from Manchester to join with the research group of Professor Zanconati. At the Metropolitan University of Manchester, she has worked at the School of Healthcare Science, Vascular Cell Molecular Biology, and at the Institute for Biomedical Research into Human Movement and Health. At Manchester University, she has studied molecular cellular mechanisms of muscular atrophy in chronic and acute diseases. She is author of 27 peer-reviewed papers. Nowadays, as component of the research group of Professor Zanconati, she is investigating some molecular aspect of prostate cancer. I think there is anything else to add? We can start. Okay. Now I gently ask to Dr. Filipa Vieira to introduce to us her lecture on histone modification in prostate cancer. Thank you very much.
Good afternoon to everyone. Well, I came from School of Allied Science in Oporto, and today I will present my work on my PhD thesis that was developed in Portuguese Oncology Institute, also in Porto. The theme is Histone Methylases and demethylases, and specifically the role of SMYD3, that is a methylase, in prostate carcinogenesis. So, prostate cancer is the second most frequent cancer in men, and most of the cases are registered in developed countries, where prostate cancer is the most frequent and the fifth leading cause of death in men by cancer. This is mainly because of the widespread of the PSA technique and subsequent biopsy. Actually, the management of prostate cancer starts with the screening of the men by evaluating mainly PSA levels, and when the values are not normal, the patients are submitted to a biopsy and then the diagnosis is performed. In diagnosis, the Gleason score is evaluated, and together with TNM staging, the patients are choose the treatment more adequate to each patient. The treatments can be just surveillance of the patients or more radical treatments like radical prostatectomy or radiotherapies, or in cases of advanced disease, only therapies that block androgens. We have a big problem concerning management of prostate cancer, that is, patients are frequently overdiagnosed and overtreated and submitted to unnecessary treatments that are very associated with some secondary effects. So we also know that prostate cancer arises to an accumulation of genetic and epigenetic alterations, and the first lesion that appears is prostatic intraepithelial neoplasia, named PIN, and then these lesions can evolve to adenocarcinomas and then to a metastatic disease. Knowing that PCA is frequently asymptomatic in these early stages, that are the stages where the treatment is more likely to be curable, and there is a difficulty in identifying the most aggressive tumors, we need to better comprehend the biology of prostate cancer and to identify new biomarkers from diagnostic and prognostic evaluation. In this field, epigenetic mechanisms can be and help to identify those biomarkers. And what is epigenetics? Epigenetics is an area that studies alterations that lead to alterations in gene expression, but these alterations do not necessarily mean that we have an alteration in DNA molecule. Epigenetics comprehend three mainly mechanisms that are RNA interference, that are small RNAs that can block mRNA and then block its transition to protein. That is DNA methylation, that is the addition of methyl groups to DNA and then block the transcription machinery and then block the transcription of the genes. And today I will focus my presentation in Histone modifications that can lead to an active or repressive chromatin and thus lead to active or repressive transcription of the genes. These three mechanisms act together and not isolately in promoting or repressing gene transcription. And what are the stones? Stones are proteins that are present in our nucleosomes, nucleosomes that have 8 stones and a single molecule of DNA. And sometimes these stones have projections to the exterior of the nucleosome that are submitted to several alterations. These alterations can be citronylation, butynylation, ubiquitinylation, but the most studied are methylation and acetylation. Concerning, for example, methylation, there are some enzymes that are responsible to promote the addition of these groups, these methyl groups, that are the Histone Methylases or methyltransferases, and there are other enzymes that remove the same groups. And another group of proteins are the proteins that can have the ability to read those marks and thus signalizing the machinery of the transcription. There are several enzymes that have already been described in literature, and the marks that we are used to study are mainly in histone 3 and histone 4. Concerning histone methylation, there are about 50 histone methylases described and more than 25 demethylases, but we don't know much about the great majority of these enzymes. Concerning histone methylation, its role is not simple. It can lead to active transcription or to repressive transcription. It depends on the residue and the state of the methylation. For example, if you look at lysine 4, that is this one, if you have a trimethylation, we have an active status of the transcription. However, if you have trimethylation of lysine 9, for example, you have a repressive state of transcription. So different marks are associated with different states of transcription of the genes. Our normal cells are associated with some specific marks, and when this modification pattern is altered, we can lead to activation of oncogenes or to the repression of tumor suppressor genes, and thus leading to prostate carcinogenesis or carcinogenesis in general. So the first part of our study was to identify which enzymes concerning the methylation and the demetylases are relevant for prostate carcinogenesis and how their assessment might provide novel tools for patient management. So we identify which enzymes are altered in tumor tissues, comparing with normal ones, then evaluate its expression and correlate it with clinical pathological data. And lastly, we assess its usefulness in clinical challenge to discriminate prostate tumor from normal tissues or to evaluate prognostic markers. We start to screen 37 methylases and 20 demethylases. We use frozen tissue from prostate tumors and from normal prostate samples. And then the results are this that I explain here. As red, you can see the genes that present an expression higher in tumors than in normal tissue, and in green, the genes that have a lower expression level in tumors when comparing with the normal tissue. We choose some of those genes to validate, and this validation was performed in a large series. In this time, we use 150 tumors and 50 normal tissues. We were able to validate our first approach, and we identify some genes that are upregulated, some genes that are downregulated. Today I choose only one to explain to you, that is SMYD3. SMYD3 presents a higher level of expression in tumors when comparing with normal tissues, and when we analyze this expression concerning PT stage, we observe that higher levels are observed in more indifferentiated tumors and more aggressive tumors. Then we try to see if SMYD3 was associated with survival of these patients. So we have samples from patients that were submitted to radical prostatectomy, and we made a follow-up of those patients and try to link if the patients that have a shorter time to a relapse, to a recurrency of the disease, were the ones that present the higher or the lower expression levels. And we conclude that the patients that have higher levels of SMYD3 were the first to have a biochemical recurrency. Moreover, we also identify that SMYD3 was able to independently predict prognosis of this disease. Taking into account these first conclusions, we observed that SMYD3 was overexpressed in prostate samples, that higher levels were observed in more advanced tumors, that this enzyme was associated with a shorter time to relapse, and also that is capable of independently predict prognosis. We decide to move on and to understand more about this enzyme. Making a little bit of introduction to this gene, SMYD3 was described as an enzyme with ability to de-3-methylate lysine 4 of histone 3, but actually we know that is capable of methylate other marks that are lysine 5 of histone 4 and lysine 20 of histone 4-2, but also other proteins that are not only histones like VEGFR. Some targets are already appointed, but little is known about the most majority of them. Concerning other tumor models, SMYD3 is frequently upregulated in other cancer models like breast, colorectal, hepatocarcinoma, and interestingly, SMYD3 was associated with transcription dependent of estrogen receptor, pointing out that this enzyme could be also associated with other models that are dependent on hormones, like the prostate model that is highly dependent of androgens. So, in the second part of the work, we intend to investigate SMYD3 function in prostate carcinogenesis using in vitro and in vivo cell assays. And for that, we evaluate SMYD3 silencing in malignant phenotype of PCA cells, identify molecular pathways and putative target genes, and determine the importance of SMYD3 methylase activity in modulation of those cells. First, we evaluate expression levels of SMYD3 in the cell lines that we have available in our lab. The cell lines are divided into groups, the ones that respond to androgens and the ones that not. And we choose one of each group to perform the knockdown of this gene. The knockdown was performed with lentiviral particles, and the silencing was confirmed in mRNA and protein levels. So then we ask if SMYD3 was able to alter the malignant phenotype of these cells. And for that, we evaluate viability, apoptosis, migration, and invasion of those cells without the expression of this gene. The first assay was MTT assay. This assay evaluates the mitochondrial activity of the cells. Only the viable cells have mitochondrial activity, and these are the only cells that are capable of alter this compound that is MTT and to promote the formation of formazan that is in this color like purple. So we can observe that although in LNCAP we didn't observe any alterations, PC3 we observe a decrease in viability when we do not have the expression of SMYD3. In apoptosis, we use a dye that marks the cells that undergo apoptosis, and we were able to see that in both cell lines there was an increase of apoptotic levels. Then we try to evaluate the migration of those cells, always comparing the cells with expression of SMYD3 and the cells without expression of SMYD3. This assay, we see the cells on the well, and we let the cells undergo it until they reach the 100% of confluence. Then we made a wand that is made with a tip of a pipette, and this is something like that, and we measure with an informatic software, we measure this area. And then along the time we will measure if this area, what is the value of this area, and to compare which are the cells that migrate more quickly. We were able to conclude that the cells that not have the expression of SMYD3 tend to need more time to complete all the area. To conclude these in vitro assays, we tend to evaluate the invasion. we use something like a mini basket that we put in the wells of our plates, and we seed our cells in that membrane, and we evaluate the cells that have the ability to degrade that membrane and to attach to the lower part of that membrane. In the end of the assay, we remove the basket, we cut the membrane, and we fix the membrane and stain and count all the cells that were able to attach the lower part of that membrane. The result is something like that you can see here, and we count each one of the cells, and we were able to conclude that we have a decrease in the invasion only in PC3 cells that are the most aggressive cells. Then we perform a very cute assay that is an in vivo assay that is performed in chick embryos, in the membrane, chorioallantoid membrane of chick embryos, in order to evaluate tumor formation and angiogenesis. Quickly, these assays, we incubate those eggs, and after 3 days, we we made a little window on those eggs, something like that, and we observe that if the eggs are undergrowing or not, and I don't know if you can see, but here we can see the embryo of the chicks. Then we close again these eggs with a tape, and at day 10 we put something like a ring, and we put our cells there in those rings in order to the cells maybe in a short space. And then after some days we will evaluate the tumors that those cells were able to formate. And the results are something like that. We take pictures in the egg, and then we remove those membranes and fix and submit it to the histological processing normally, and we observe those membranes under the microscope and measure tumors that are formed. Is in a bigger magnification, we were able to conclude that is a trend to tumors with a lower size in the cells that do not have the expression of SMYD3. As you can see here, a big tumor and no big tumor in the lower parts. Then we ask which genes are being regulated by SMYD3, and we perform an assay that compares transcript levels of several genes involved in several pathways like cell cycle, apoptosis, DNA repair, and some of the pathways that are frequently altered in prostate cancer. This is the result, and I will highlight one of the genes that was overexpressed after silencing of SMYD3, that was cyclin D2. Cyclin D2 is a gene that is involved in cell cycle, although the role of this gene as a tumor suppressor gene or an oncogene is not clearly understood. In prostate model, there was already been published that this gene is frequently downregulated in prostate tumors when comparing with normal tissues, pointing out that this gene must be a tumor suppressor gene in this model. So we intend to evaluate if the marks that the enzyme is responsible for, that histone marks that I told you about, are present in the promoter gene of cyclin D2. And we are able to see that was a decrease in one of the marks that we study, that is the trimethylation of lysine 20 of histone 4. This mark is a repressive mark, and we have a decrease of this mark after silencing that maybe is responsible for the overexpression of this gene that I showed you some slides ago. Interestingly, when we evaluate in the tumors that we have in the lab, we see that SMYD3 presents higher levels in those tumors, where cyclin D2 presents lower levels in the same tumors. And to finish, we also aim to identify if those alterations that are caused by SMYD3 are associated with its activity as methyltransferase. So we searched about the sequences of this protein, and we identify a region that is responsible for the activity as an enzyme, as a methyltransferase, that is the set domain, and we decide to delete two regions that are present in this set domain. By direct mutagenesis, we delete those regions, we transfect into bacterials to have a lot of amount of DNA, and then we transfect our cells that are silenced for SMYD3. As a result, we were able to see that the cells that were transfected with the mutant forms of SMYD3 have a similar result comparing with the cells that do not have the expression of SMYD3, pointing out that the results that we obtained were related to its activity as methyltransferase. As a conclusion, SMYD3 expression levels were associated with aggressiveness and predict disease-specific survival in clinical localized PCI patients that were submitted to radical prostatectomy. SMYD3 proved to be associated with a more malignant phenotype in PCI cell lines, supporting its oncogenic role, and might be associated with an increased tumor in vivo. As a target, we identify cyclin D2 as a target of SMYD3, and that regulation is done through its repressive mark, that is trimethylation of lysine 20 of histone 4, and its oncogenic properties were related to its methylases activity, mainly on one of the domains that we study. So, as a final remark, and knowing that this information, we point SMYD3 as an oncogene in PCA, and that might be an attractive target for personalized therapy for patients that have prostate cancer. As future studies, we intend to evaluate the expression of SMYD3 in prostate biopsies to see if they can help to adequate the treatment of those patients, and to identify other genes that may be targeted by SMYD3, and also to explore the role of others methylases and demethylases in prostate cancer. And finally, I want to thank the team that worked with me from Portuguese Oncology Institute and from School of Allied Science, and that's all. Any question?
Lecture. Thank you very much. Some question?
We need the ice cream.
Okay, I stay here. So, no, the work is really very interesting. I have two